Hey guys, today I had the pleasure of talking to Sara Rivero-Calle. She's a researcher with the Skidaway Institute in Georgia, working with coral reef ecology and continued innovation on remote sensing tech. Remote sensing is the use of high-resolution satellites or aerial images combined with algorithms and statistical modeling to uncover amazing information about the world. So, for example, you can use this tech to track the amount of a certain phytoplankton in the world and like across the water based on how much of a certain light wavelength was returned to the satellite or based off how that light was breaking apart and making an algorithm to track that. She's also involved with amazing work on CubeSats, which are mini satellites, usually about 10 centimeters by 10 centimeters cubed. And actually on a previous project of hers, their satellite was one of the few selected by SpaceX to take up and bring into orbit with their first payload of commercial satellites, which is insane. I had the best time talking to her, and she did an incredible job breaking down many of the concepts we talked about. We'll definitely be hearing more from the Skidaway Institute soon. Enjoy, guys.
Hi, so my name is Sara. I'm originally from Spain, and I grew up in Madrid, in a big city, 4 million people, and it was 3 or 4 hours away from the ocean. So for me, going to the ocean was a big party, a big event. We would usually only go there once a year. So I think that was one of the reasons why I was always fascinated by the ocean. And besides that, I've always been interested in nature and why things are the way they are, the little details. My grandpa he did, I don't know what it's called in the US, but he was kind of like a nurse. And but he told me that he was a really bad student when he was younger, and he would just play hook. How do you call it? Play
Play
hooky.
hooky, like skip
Play
class.
hooky.
Yeah.
And go, and he knew all the birds, all the mushrooms, all the trees. It was something that really fascinated me, how much he knew, and I was always like, oh, grandpa will know this.
Mm.
Which is also interesting because now kids think that Google knows everything. For me, it was my
Yeah.
grandpa. um, so yeah, so I was always interested in biology and nature and why the things are the way they are. And so I knew from a very early age that I wanted to be a biologist. That's what I wanted to do. Um, and yeah, so in Spain, it's very common to stay with your parents throughout college. And being in Madrid, I had really good options of public universities. And so I was staying with my parents and studied general biology. But there was something in me that was saying, I really want to study the ocean, and I'm not getting the classes that I need here.
Mhm.
And in Europe, we have this exchange program called Erasmus, which is like a study abroad program. And so I took that opportunity in my fourth year. It's 5 years to go through college in Spain. So on my fourth year, I won a fellowship and I went to Italy, where they had a really good program in marine science. And that's where I first really started taking classes in marine science. But throughout college, I always did a lot of volunteering work. I did work with orcas in southern Spain. We didn't see any, but I spent a whole week
I was gonna
just
say there are a
staring
lot
at
of
the
work ethics
ocean.
over in yeah.
We were told, yeah, you see them at the Strait of Gibraltar. I saw pictures and I was like so motivated. I'm gonna see an orca.
That's so
But I spent a whole week and I didn't see any. Um, I also spent a month in Greece working with sea turtles in marine protected areas, and I saw the hatchlings. So I did some work with, I joined this club for marine mammals
Mhm.
studies, and we organized a lot of workshops and seminars. We brought speakers in, so I was very involved with anything marine related. Um, and then after my experience in Italy, where I learned a lot more, I learned about marine biology, I learned about marine protected areas. I started scuba diving,
That's so cool.
and a
That's
lot of
I can't wait.
research.
Sorry.
Yeah, Yeah. So, and they had a class on marine photography, underwater photography.
Hmm.
And I took that, and the professor said, well, if any of you are interested in working in my lab, I have some projects and some graduate students who might be, um, who could use your help. So I went and talked to the professor and I said, I'm interested. I want to do this and I need to get some credit in research for my undergraduate thesis. And he said, yeah, come on in. And so I started working on a project of underwater photography and photo transects in the Red Sea. And I started learning about coral reefs. And I had some really good professors who were experts in sponges in that university in Genoa. And so I thought, well, this is a great idea. Maybe this is what I want to do for grad school.
Hmm.
You know, coral reefs are in the tropics. That sounds perfect for work. Uh,
Yeah.
Corals don't move, so it's much easier than birds or marine mammals.
Hmm.
And there's a big economic impact related with coral reefs. So I decided that that's what I wanted to do. So I went back to Spain after that. I took a bunch of classes in Italy, got really good grades. My dad was hesitant when I was going to Italy. He said, "I don't want to pay for you to go party in Italy because that's what people think people do during Erasmus, which a lot of
Oh
people
yeah.
do." And I partied a lot, but I also studied a lot.
Yeah.
When I
You
came
gotta
back
do
with those
both.
grades,
Yeah.
my dad was like, "Oh, can you stay there longer so that you get a better GPA?" And I was like, "Oh, really? Now you want me to stay?"
Yeah.
Um, but I preferred the method in Spain, and I went back and I decided to finish my degree in general biology and then look for grad school programs in marine biology. And
So what
because
do you
of
mean by the
that,
method?
I'm sorry?
You said you prefer the method in Spain. What do you mean by that?
Yes, the Spanish education had a lot more lab work,
Uh-huh.
and the way things are taught are more hands-on and more applied. In Italy, I was amazed that you would just go and take the exam. They were all
Yeah.
oral exams, take them in front of the class, everybody's listening, and you have to
Yeah.
like recite like a parakeet,
Yeah.
whatever
Or
the
like
professor
memorization
taught you.
or theoretical, but not hands-on.
Yeah, it was very memory based. And one thing that was really good from that is that I really enhanced my memory skills, and that I learned how to express myself better, because in Spain it's the opposite, it's all written. um, so I think that Italian students in general are really good speakers because of that.
Hmm.
But I didn't like the way they taught us much. Just to give you an example, we were all year round talking about these seagrass beds in the Mediterranean Sea.
Hmm.
And there's this species called Posidonia, and it's an umbrella species that is like the forest of those seagrass beds. It's what conforms the structure for all the other organisms to live in. And they are very worried because they're declining in the Mediterranean Sea. That has a lot of ecological consequences. And like I said, we were talking about this all year round. My friend got straight A's. I got really good grades. We were after classes hanging out at the beach. And then she points at this green thing on the beach and she's like, "Oh, I wonder what this is." And I was like, "This is Posidonia. I can't believe you don't even know how it looks like."
Yeah.
So that was an eye opening for me. I was like, no, this
Yeah.
system doesn't work well.
Yeah.
But I did, because I was coming from another background, I knew what I wanted to do, and I tried to get as many opportunities to do stuff as I could. So,
Hmm.
for example, the scuba diving class, for international students, we weren't supposed to be taking it because it was expensive and the department was paying for it, and they don't have a lot of money in
Hmm.
Italian universities. But we convinced them, and I was able to take the classes, me and my friend. Um, we also went on a research cruise. Like I said, I worked at a lab. Like I made the most out of it, but not
Mhm.
everybody does. So if you just stick to the basics, you're not going to get that holistic education. But overall, it was fantastic. I learned the language. I learned a lot about marine sciences and made lots of friends. I'm still friends with them 20 years later. Um, so it was a really good experience, and it was the first time I actually lived abroad,
Mhm.
and away from my parents, and I was 21, 22 at the time.
Mhm.
So it was a big deal.
Yeah.
We all talk about the post Erasmus depression, but
Oh
the
yeah.
reason why I'm mentioning this is because that's how I started going towards marine sciences. And I started thinking about what I wanted to do next. So I was analyzing, like I said, photo transects of coral reefs from the Red
What's the
Sea. I never.
transect?
Okay, so in ecology, we often, one of the ways in which we sample is we, especially in coral reefs or in land, on land, you just set up a tape measure, um, and that's your transect. And along that transect, that tape measure, you look at specific quadrats, which are actually squares. And so you go and place a square, say every meter, and
Mhm.
you look at what's under that square. So you can say, okay, there's so much Posidonia, and you can look at it in terms of counting or percentages. So you
Mhm.
can say, there's so much Posidonia, there's so many sponges, so many crabs, so many, I don't know, whatever, clams, whatever you see in there. And so you start counting it. Um, and then you do several transects and you look at averages. And so you can say, in general, in southern Italy, for example, we see that there's a percentage of 70% Posidonia, 20% sand. Okay.
Mhm.
So then you go 10 years later, you do the same transect, and you find that there's only 50% Posidonia and 50% sand. So you're starting to see a decrease there. You can also do this with coral reefs, and you can say how much of the coral is alive, how much is out. There's a competition between algae, corals, and sponges, for example, in terms of substrate,
Yeah.
which is the surface on which they can start to grow. They need something to grow upon. So there's this competition for space, and it's very intense in coral reefs. So that's one of the ways in which we can measure the health of coral reefs or seagrass beds and things like that.
So a transect is like the ecology world's survey system, kind of.
Yeah,
Is or a
exactly.
method? Is a
Survey
transect a
system.
set length? Like the unit? Like is it like
It
a like?
depends. You can define, as long as you define it,
Uh-huh.
it can be
They
whatever
can
you
convert
want.
it,
So
yeah.
it
So
could,
that they can convert it at least. Yeah.
depending on what you're look. So if you're looking at changes in the cover of Posidonia,
Hmm.
if you only do 10 meters, it might not be enough. Maybe 100 meters makes more sense. But remember, you're doing this underwater, so you have to
Hmm.
be scuba diving and
Yeah.
counting and whatnot. Um, what I analyzed was somebody was scuba diving, and rather than sitting there with a, well, not literally sitting, but underwater, being with a clipboard and making notes of what they're seeing, you can go and take a picture of each of those quadrats along the transect, and that's much faster because it's just a picture.
Yeah.
Boom, next, boom, next. And then you go back to the lab and analyze it. So somebody went out there, took those video or photo transects, and then came back to the lab. And so that was a lot of work to go through, but it gives you a better quantitative idea of whatever you're looking at. If you're looking at, for example, what are the richness or diversity of corals in the Red Sea? So how many species, how much of each species, and so forth. So there was a grad student working on that. She was my mentor at the time, and just that she jokingly said, "You're to say la mia schiava," she said, "You're my little slave." And that sounds really bad, but she just said it jokingly because she knows it's a lot of work.
Yeah.
And so I was sitting there over a whole month, and that's all I did for 8 hours a day.
Yeah.
um, but I stayed an extra month in Italy in, I think it was June, to complete this work. And then over the summer, I did a write up, and then the graduate student who was my mentor, she was very excited and very happy with my work, and so she invited me to be a co-author in a poster, which
Oh, that's
is like the
awesome.
biggest thing. Yeah, so in Europe, we don't have what they call here in the US REUs, research educational research experiences. I'll look it up. I don't remember,
Uh-huh.
but they have this thing called REUs, which is an opportunity for undergrads. Maybe the U is for undergrads. Um, it's an opportunity for undergraduates to go and for the summer work on a real world research project, and so they learn a lot of the skills that you would need to become a professional researcher.
Hmm.
So that's really valuable. And so in Spain or in general in Europe, that is not a thing. And most of these opportunities are on a volunteer basis, or because you know a professor, you've been taking his or her class, and you say, "Yeah, I'd like to work with you." And so you
Hmm.
often just volunteer. It's not paid work, unfortunately. But that all those volunteering opportunities really helped me figure out what I was good at, what I liked doing, what I saw myself doing. And I always highly encourage all the students I encounter to do these kind of experiences.
Hmm.
Um, yeah, so that experience led me to Puerto Rico, because remember I was interested in coral reefs,
Yeah.
and so my undergraduate university had an exchange program for graduate school between my university, Universidad Complutense de Madrid, and the University of Puerto Rico. And I really liked that program because it was set up on a small island surrounded by coral reefs
Hmm.
with iguanas running around. That's where our lab and our classes and everything took place. And B, they had a bilingual program, Spanish, English.
Hmm.
So even though I had a good level of English before I went there, my English was not technical.
Yeah, he
So
would have let you
that
sit
was a
in
really
too.
good opportunity.
Hmm.
So I went there initially for a year. I was 24. I was experiencing everything. I wanted to do everything. And so I went there with the idea of going for a year. And if I liked it, if I could find funding to stay there longer, I might stay and do a master's or a PhD. And if I didn't, well, it was a year abroad in a really
Hmm.
cool place, and you can't take that away from you. So I went there, and I started. There were a few professors who were doing coral reef ecology, and I ended up working with Roy Armstrong, who does a lot of satellite remote sensing. So he uses satellite to understand ocean ecology, and he started talking to us about what we call the ocean color. And he actually had, and I told myself I was not going to do any more video transects. He had all these really cool video transects from deep coral reefs, the ones that you can't get to with recreational scuba diving. He had this robot that would go underwater at between 30 and 100, 150 meters depth and do those transects I was telling you about with video, um, and collect the data, and then somebody had to look at them, right? And I was interested in continuing my work on coral reefs, and in particular sponges. And because they were deeper coral reefs, and I had found out that well, when I got to Puerto Rico, there was this big coral bleaching event, one of the largest in a really long time. It was 2005. So I was fascinated when I started
What
scuba
is
diving.
coral bleaching?
Oh, okay. So coral bleaching is this process by which there's a disruption in this symbiotic, this mutual beneficial relationship between corals and some microalgae that live inside the coral tissue.
Mm-hmm.
So, like I said, it's mutually beneficial. So both the coral gets something out of it, and the microalgae get something out of it. But when the conditions are not right, the coral or the algae decides to break up this relationship. And so that microalgae has some pigments, some coloring substances in it, that will give the color to the coral, because the coral itself is actually transparent.
Mm-hmm.
So when there's this disruption and there's no algae in the corals being transparent, what you see is just the substrate, which is usually white. So
Mm-hmm.
what you see is where the coral is sitting, and so it looks white. So it's like
That's really
as
cool.
it's as if you see that snow, it snowed all over the coral reef. They're
Yeah.
usually brownish, yellowish. In the Caribbean, there can be some really interesting colors in the Pacific, but in the Caribbean, where I was, it's brownish, yellowish, and so suddenly you see this whole white blob covering the
Yeah.
rocks. It's
Yeah,
really
that's so wild.
impressive to
I
scuba
never knew that
dive.
algae was what gave coral its color either, which is so how does it just leave? By leave, do you mean like literally like detaches and goes flees or like dies off for some reason in the coral? Or do we not know?
So I haven't really kept up with the literature, but when I got there, because I was so amazed by it, I read a lot about it, and we don't really know whether it's the coral is spilling it or the algae leaving or a
Hmm.
mixture of the two. But one of the triggers that could cause this coral bleaching is excessive temperatures. We know that when they're above 30, 31 degrees Celsius, there's bleaching.
Hmm.
So if you put this in the context of global warming,
Hmm.
we're seeing that there's more and more bleaching events. And if you remember, when I was saying that it's mutually beneficial for both of them to be together, it's because the algae produces some byproducts, some like digestion results,
Hmm.
that is the food for the coral.
Hmm.
The coral is, we could say, carnivorous, so it's eating food from the environment, but it's also eating whatever the algae doesn't want.
Hmm.
So if the algae goes, there's less food for them, they're stressed out, they have to think about how they use their energy and their resources. So for example, it could um, affect reproduction, it could affect them in response to diseases. So they
Hmm.
might be more prone to getting sick and dying. So that's why we're very worried with coral bleaching, because when they're white, when they're bleached, they're a lot, they're weak, and they're more sensitive to any other stressors. So it's very easy for them to then follow up with a disease and then die.
Hmm.
And if they die, it's really hard to recover from that. Like I said, there's this intense competition for space in coral reefs, because there's a lot of organisms trying to find a place to sit and grow.
So would
So if
coral
the
be considered,
coral grows,
sorry to interrupt you, would the coral be considered you mentioned an umbrella species before. Is coral considered that because it provides like an ecosystem for a lot of other species? Okay.
yep. Yeah, because as they grow, they also build more substrate, they
Hmm.
build more rocks underneath it. So that allows for other organisms to grow. So if the coral dies, then the sponges and the algae say, haha, we can take over that space.
Yeah.
And they grow a lot faster than corals. So, especially algae. So, that's one thing that is very well documented, for example, in Jamaica. I think it's Discovery Bay. That's like the example that everybody
Mhm.
talks about in class. They removed a lot. They were overfishing, so they removed a lot of the fish that were eating, preying on the algae. So then suddenly there was no control over the algae. The algae started overgrowing the corals, and then you add to that, nutrients from runoff that will make the algae grow faster,
Yeah.
and changes in turbidity, the water becoming less clear, and the algae that live inside the corals need that light, that sunlight, to do the photosynthesis and produce those products that the corals are eating. So it's all
Yeah.
intertwined.
Super entwined. Yeah.
Yeah, and then
So
lots of
interesting.
tourists coming in, and as much as we like to go, scuba diving, we're sometimes don't control very well our flippers, our
Mhm.
our fins, and or we just like step on the corals, and then you're making a really big damage to the coral.
Mhm.
Um, anyway, so I was fascinated by coral bleaching, and I found out that there's actually also sponges that have a similar relationship with algae inside of them, or intertwined in their tissues. So, because I was handed this amazing amount of data of the deeper coral reefs where there's less sunlight, I was interested in seeing how light might be affecting effects of how turbidity might be affecting the amount of light that corals and sponges would be having at those depths, and seeing if there was any relationship between those. So for my master's degree thesis, I did a study, first of all, just documenting what was there,
Mhm.
because it was in the early 2000s when we started thinking or finding these what we called mesophotic. So meso means medium, photic means is related to light. So medium light ecosystems for corals to grow. We knew about the ones that were shallow that we could scuba dive to, and we know about the really deep ones, but it turned out that there was this in between, this mesophotic system, where we were seeing amazing ranges of coral cover. And so we started thinking that maybe those could help um, as nurseries for the shallow endangered coral reefs. So there was base, and because they were difficult to access, you needed either a robot or to get a very highly specific scuba diving training to get there. We basically didn't have any information on these mesophotic coral ecosystems.
Mhm.
So our lab and the groups in the different groups in Puerto Rico, there were some groups who were studying the algae in mesophotic ecosystems. There were some people studying the sponges, the corals, um, and with different approaches. Some people were using DNA, some people were using the transects, some people were um, it was a combination. We were one of the few centers in the world who had access to those mesophotic reefs, and who were starting to document these around
Mhm.
Puerto Rico. But our lab was the only one who was using the robots to do the transects. So we were able to cover a lot more spatial extent. We had information from different parts around Puerto Rico. And so my thesis was, first of all, just documenting what was the coral cover and whether there was a relationship with depth. Do we see an increase or a decrease in corals and sponges with depth? And this is actually something that is still debated in 2020. There's been two groups that are still discussing this, and it was really neat that they were still citing my master's degree.
Yeah, that's
uh,
really cool. Yeah.
yeah,
People
so
are
that was the first
your
time I
work.
was like, oh, yeah, my work is actually interesting, precisely because I was one of the first people who started looking at the sponges, because everybody was so focused on the corals. I was interested in the sponges.
In that mesophotic level,
Mm-hmm.
like in that environment. Mhm.
Yeah. So, but like I said, my advisor, he did all this cool stuff with the robots because he used to spend summers at Woods Hole Oceanographic Institution in Massachusetts. And he started talking to engineers there, and they came up with this project together. But his main thing is satellite remote sensing. So looking at, I think his thesis back in the 70s or so was in looking at changes in the mangroves in Puerto Rico,
Mhm.
and he started using aerial photography. And then as we started getting satellites in the 70s, 80s, he started incorporating that. So I took a satellite remote sensing course from him, and I started realizing that, it takes so much work to just look at one coral reef. How are we going to deal with global problems? It's a no-brainer. We need to use some other system
Mhm.
that can give us a global snapshot, and that's satellites. And I just became amazed by how little we actually knew and how much was still to be done. And I thought that that would be a really good way to go for my career.
Mhm.
Um, and because at the time there was a lot of talk about ocean acidification and the relationship between corals and phytoplankton. By phytoplankton, I'm referring to the plankton component, so the living organisms that are floating in the water, but that do photosynthesis. That's what the phyto stands for. Phyto
Mhm.
means plant. So they're plant
Diet.
plankton. And I'm interested in, uh,
Oh, I said like
what did
diatoms,
you say? I'm sorry.
like diatoms. They're
Exactly, like
for
diatoms. Yeah, yeah, so diatoms are phytoplankton, but
Mhm.
we also have the animal plankton, the zooplankton, and that's the one that people immediately think of as like whale food. Oh yeah, the krill, the plankton, that's what
Yeah.
they eat. But I'm interested in the ones that are plants because they do something unique. They
Mhm.
do photosynthesis.
Yeah.
And a lot of people don't realize this, but half of the photosynthesis on the planet is done in the ocean.
Mhm.
And these micro plants are the ones responsible for this. So,
So is that a combination of the algae and the phytoplankton that do like the majority of that photosynthesis in the oceans?
yeah.
Okay.
Um, the neat thing about these microscopic plants, phytoplankton, is that they reproduce really fast. They're like bacteria. So they can just split in two in a day or several times a day, depending on the species, or it might take 3, 4 days. But the rate at which they reproduce is much faster than Posidonia could go,
Mhm.
you know? So, and they're everywhere. And they are a big part of what gives the color of the ocean that we can see from satellites. So that's why we talk about the ocean color satellite remote sensing.
Mhm.
And that's what my expertise is in now. So it's all sort of related, but
Yeah, that's pretty
it
cool.
took a turn. And the reason why I started getting interested in this is because I started hearing about a particular type of phytoplankton, of micro plants, um, that actually cover themselves in calcium carbonate shells. They surround themselves by little platelets, and calcium carbonate is like chalk and it's white, right?
Mhm.
So it's the same thing as what the corals secrete. That's what they make their substratum, the rock in which they sit upon. So there's the relationship there between corals and phytoplankton,
Mhm.
specifically this group of calcium carbonate forming phytoplankton, which we call coccolithophores. And it's for some people it's a tongue twister, so I like to break it down. So if you break it down into the first part of coccolithophores, it's coccos. Coccus means sphere, like Synacoccus, coco, like there's a lot of bacteria that are rounded
Mhm.
that are spherical, and so that's where the name comes from. So they're spherical. Lithos is for rock because they cover themselves in this calcium carbonate rock. And then force means that they bear. So they're spheres bearing rock. That's the
Okay.
translation. Coccolithophore.
Coccolithophores. That's so cool.
Now
Yeah,
you'll
I
get it.
never knew because I've heard it before, but I never took the time to like break it down, and it definitely helps you remember it. Caucasus
Yeah.
sphere, lithos, yeah, earth,
Yeah.
or because it's carbonate.
So So if you're familiar with ocean acidification, what it means is that all this CO2 that we're producing in the planet, there's some that's being I mean, when you think about all the pollution and all the cars and industry, we're producing lots of CO2. It's going to the atmosphere, but
Hmm.
it doesn't just stay there because of the natural chemistry of the Earth. It's going to try to equilibrate with the ocean.
Hmm.
So all that CO2 starts going into the ocean, and that's like carbonating your soda, same
Yeah.
idea.
And that's carbonic acid, right? What it stores in the oceans as.
So you start having more CO2 in the ocean, and that changes the pH. And what it does is it makes it more acidic. If you have something acidic and you mix it with calcium carbonate, it starts bubbling and dissolving. Um, so we're worried that anything that's made out of calcium carbonate, which there's tons of things in the ocean, we're talking about coccolithophores and corals, but lots of seafood that make shells and fish, like a lot of our seafood industry could be affected. That's why a lot of scientists are worried about ocean acidification.
Hmm. So
Um,
does is it literally like the more acidic water that has more like carbonic acid in it? It's pulling the calcium carbonate that these things are using to make their shells and everything. It's pulling them out of them and into the water, like it's pulling the calcium carbonate off.
it's just making it harder for them to produce the calcium carbonate
Okay.
because it does it's there's more hydrogen ions in the water. That's
Hmm.
what makes it more acidic, and you have to combine that with the carbonate ions. And anyways, it's a complicated thing to
Hmm.
explain. I'm really not good at doing this.
Oh no, you're fine. Oh yeah, it's
Podcast,
it's,
you'll
it's
have
super
to edit this
interesting. Yeah,
part.
you definitely don't need to answer all the questions I'll ask, but I'm
Yeah.
happy you'll be giving it your best. But yeah, that's super interesting.
But yeah, so I started becoming interested in this phytoplankton because they're covered in calcium carbonate, the microscopic, they're phytoplankton, and because they're so white when they bloom, we meaning like they're very happy and they start reproducing a lot and they start blooming in the ocean, and it looks like as if someone dropped milk in the water.
Really? That's wild. Yeah.
Because they're white, remember? So it's like dumping milk in the water, and when there's many, many of them, we can actually see them from space.
That's so cool. Yeah.
So I was wondering, okay, so what's going to happen? That's our lab rat for ocean acidification in phytoplankton, and
So the less white we see, the worse, because that means there's like.
well, we don't really know, and this is a very anthropogenic way of thinking about nature. We don't know why they're there.
Mhm,
What's their function, right? Nothing has a function; they're just there.
mhm.
But so if we see that there's a decrease in them, we might think that that's a symptom of ocean acidification.
Okay. It's a better way to put
Now,
it for
we
sure.
don't know
Yeah.
but we don't know very well who eats
Yeah.
them, so we don't know the ecological consequences of seeing a decrease in coccolithophores. But we do know that in the past there were lots of them, so much so that when you go to southern England, you may have heard about the White Cliffs of Dover.
I think I have, yeah, but I never knew it was tied into this, but that makes sense.
That is an accumulation of the shells of the
Mhm.
coccolithophores over time that died and sunk to the bottom, those cliffs are made of microscopic coccolithophore shells.
That's so cool.
So yeah, when you think about it, it's like,
Yeah.
whoa, so this thing is 6 microns or so in diameter. Well, there's different species, but the typical one that causes the blooms that we can see from space, or even with a naked eye if you're out on the boat, they're 6 microns. So that's 10 to the minus 6 meters of
Uh-huh.
a diameter. So it's like super tiny.
Yeah.
How do you get to accumulate enough to see cliffs,
Yeah.
these guys?
That it builds up so long. Yeah. So there's
Yeah,
a lot of them in the past. Yeah.
right. So that's just a testament of what has happened in the past, what could happen in the future, and how um, these microscopic organisms have such an important effect on a global scale.
Hmm.
Um, so the hypothesis was that if you increase the CO2 in the atmosphere, it's going to equilibrate with the concentrations in dissolved CO2 in the ocean. So that's going to make it more acidic. So if you're made out of chalk, calcium carbonate, you're going to start having difficulties making new shells, or you'll start dissolving if it's really dramatic. But
Hmm.
that would take a really long time. But the funny part is that for my PhD thesis, I looked at this long-term time series from the North Atlantic, where they collected data regularly since the 1960s,
Hmm.
and it's still ongoing today. And I looked at the data, and what we're seeing is not a decrease in coccolithophores, but an increase in coccolithophores.
That's interesting. And that's just it is there data scope like that's like global that there's an increase or in a certain area?
That's the thing with ocean data. It's not easy to collect. You can't go to the
Yeah.
middle of the Atlantic very easily. So there's not many time series to compare it with. This is the longest in the world, and we
Hmm.
see an increase, especially after the 90s. And so I looked at any other environmental data that I can use to compare it with the coccolithophore data, to try and see if I could figure out whether this could be natural variability kind of thing, like this is an El Nino or an Atlantic multidecadal oscillation. Is this global warming? Is this something local from that region? Is it temperature? What is it? So I use some statistical models, which people usually refer to as machine learning, but there's more than that. There were some linear models and some really complicated black box ones like neural networks. Um, and I found out that there was a model that would fit the best to my data.
Hmm.
And that was using what is called a random forest model, which is very popular right now. Lots of people are using it, but I went and compared 10 different models to see which one was best. And according to that, what I found is that the variables that were most important factors for that increase were the CO2 concentration and warming, and it's a little bit linked to this Atlantic multidecadal oscillation trend. It's like an El Nino, but it repeats on not
What
5
are
to
both what are both of those? What's an El Nino?
so an El Nino is this event that happens periodically every 5 to 7 years, more or less, and it has effects all over the world. But it is related to changes in pressure differences in the Pacific and Indian Ocean, and that leads to changes in the way the ocean circulates, and that affects rain events and flooding and droughts in different parts of the world.
Hmm.
So we know that there's a few of these. There's ones that are global, and there's some that we know are more local. Kind of like well, it's not like the monsoons, but it's something that we know that happens. But if they happen every 20 or 30 years, it's harder for us to have a record of them. But if it's every 5 to 7, it's more common. So, for example, the coral bleaching event that I was mentioning in 2005, that was linked to an ENSO event, we think.
Hmm. And so we don't
Um,
know the cause of those El Niños. We just know kind of how more like kind of how they work and when they happen, or like around how often they happen.
natural It just naturally happens. So there's one that has a longer periodicity, which is the AMO.
Hmm.
And so in the 90s, it was in the positive phase. And so that is associated with warmer temperatures. So there's some of that in there. But the CO2 was the most important factor according to my model. That's what the data showed. So I was like, well, that doesn't make sense. Why is CO2 increasing, making these guys happier?
Hmm.
And so I went back and I did a study, what we call a metadata analysis, which is when you go and look at other people's research and combine all that to get some like global picture idea of what's
Hmm.
happening, and you combine everybody's data. So I started looking at laboratory studies where people were looking at effects of CO2 in what we call growth, but it's actually the reproduction rates of coccolithophores. And so, what if you remember, coccolithophores are still phytoplankton,
Hmm.
which means they're plants, which means they do photosynthesis, which means that they take up CO2 and release oxygen.
Hmm.
So, if you give them more CO2, they
You
could
give
be, in
them more
fact,
food. Yeah.
but why was it affecting coccolithophores more than other groups is what didn't make as much sense.
Hmm.
And it turns out that the different phytoplankton groups have different thresholds and different amounts of CO2 at which they just like can't take anymore. They just get saturated. So coccolithophores, they don't get saturated as fast as diatoms, as you were mentioning. So for them, this increase in CO2 was seen as an increase in food
Hmm.
and resources, whereas for diatoms, they couldn't take anymore. They were at their maximum. So what I think is happening is that that increase in CO2 was increasing the rates of photosynthesis. So they were happy, they were growing more. But if we keep increasing the CO2 levels, it'll get to the point where they're going to be saturated, but it's going to start affecting the chemistry of the ocean and it's going to affect the calcification process for
Hmm.
coccolithophores. Again, it's a single cell and they have to decide whether they use their tiny resources to reproduce or
Or flush it
to
out, or yeah.
to the shells or what they do with it. So right now they're happy, but I think that based on this analysis of everybody's laboratory studies, what I was seeing is that they're getting to that point where they're gonna meet their maximum and they're just not gonna be favored by any more CO2 in terms of photosynthesis, but it's going to start affecting the calcification process.
Hmm.
Now, um, what does that mean in the long run? I think that maybe coccolithophores will still thrive because that's the other thing. We don't know why they actually make these platelets.
Hmm.
There's many different theories.
By platelets, you mean like the calcium carbonate shells?
Planet
Okay.
shells, yeah, which we call coccoliths. Um, we don't know why they make them in the first place.
Yeah, I almost asked that because I was like, wouldn't that hurt? Like, wouldn't that make it harder for them to like the sunlight to hit them and then do photosynthesis? Um, but yeah, that's really interesting that we don't even know why they make their shells.
We don't know, and
Um,
there's species that only make one layer, but there's other ones that keep making layers and layers. We don't know if it's for protection, if it's a byproduct of their metabolism, We don't know, we really don't know why they make them. But I have the sense that I have this hunch, but I might be wrong, that they might continue to live on after they lose their shells. But we don't know what that's going to mean for the ecosystem. We don't really know who eats them very well.
Hmm.
Um, so it's all a big unknown, and I just wish we don't get to the point where we're at such high levels of CO2 that everything is going to be disrupted. Because when we change the balance of the ocean, we don't know how all the different species are going to be affected. Some are more affected by temperature, other more for CO2, other more by nutrients. So it's just so many multiple stressors at once that it's really hard to make predictions as to what's going to happen in the future. And one of the issues is that, yeah, there has been past periods in Earth's history where we've had a more acidic ocean, but it's never happened this fast. Corals can adapt to this rate at which it's changing.
Hmm.
We don't know if the rest of the how the rest of the phytoplankton how the rest of the phytoplankton is going to um, respond to all these changes and at which rates. And what's clear is that there is going to start to be some mismatches between the predators and the prey, the quality of the prey. So which phytoplankton is going to be the winner, because they're always competing amongst each other, but we don't know which one is going to dominate. If there's going to be one, are they going to be the large ones, the small ones, new species? The harmful algal bloom species might be more predominant. So it trickles down the ecosystem to the point where we really don't know yet how that's going to be affected. And so we need to rely on models, and all models are wrong, but some of them are useful.
And it's so interesting because there's even like like ecology is such a complex thing because there's not really even like a goal you can shoot for. Is like what I was thinking about in terms of, like you said, it's all a balance of all these things in the ecosystem. So there's not really like a better or worse thing happening because one, whatever happens is better or worse for one. Like, like more calcification, right? Or more acidity right now is better for the coccolithophores, but worse for like the like diatoms. Um, but like there's no, because the natural world is going to balance it out. There's no like common goal. Like, do we want an ecosystem that's just making most oxygen? Is that the goal? Then we can shoot for certain ratios of all these things. Do we want so there's just most of this one thing? Do we want so it's easiest for fish? So there's not even like it's so hard in itself to analyze and make models, like you said, and get data and like figure out these things. But it's even harder that there's not even like it'd be so much easier if they were like if the whole world is like, all right, let's perfect every ecosystem in the whole world so it's best at making
But
oxygen,
that's
whatever.
science
Yeah, and you can't
fiction.
do that. Yeah, yeah, but it's just so wild that the
We
ecology.
just
Yeah,
we still need so much baseline data. Like I said,
yeah,
we don't know who coccolithophores or why they make the shells. There's many
yeah.
different theories, but we don't
Or
know.
yeah, or if it's good or bad that we have more of them. Yeah, yeah. That's
Or
so
not.
interesting.
And these microorganisms, they keep evolving and keep changing. So there's a few people, for example, who have been looking at some other microorganisms that I've also studied in my PhD thesis, that have the capability of living in what we call ocean deserts, in the sense that there's no food for plankton. They're what we call oligotrophic areas. And again, breaking it down, oligo means a few little, trophos means food. So areas where there's not a lot of food, like in the middle of the Pacific, there's not a lot of food. Or yeah, and so in those areas, if you're able to produce your own food by, for example, extracting nitrogen from the atmosphere and using that as your source of energy, then you're the king of the world.
Mhm.
So, for example, there's some researchers where I did my postdoc in the University of Southern California who were looking at long-term changes in this group, the trichodesmium that fixes nitrogen, and see what are the changes in their evolution when you look at many reproductions. So you can do this with microorganisms where evolution happens at a faster rate
Mhm.
um, than with seagrass or coral reefs. They have a harder time to adapt to those changes, but with microorganisms it's faster, and so they were looking at changes, long-term changes. There's just so much we don't know, and it's really hard to do, to look at global
Yeah.
problems,
So
and
interesting
maybe,
though. Yeah.
and that's the other thing, we might be using what we call strains. So you get one, for example, one coccolithophore or one trichodesmium, and they keep reproducing, and we have a line of cells that are all with the same genetic material
Mhm.
because they all evolve, they all come from this one cell. So when you look at that strain, how representative is that strain
Yeah,
from
exactly. At
the
what point is that too far removed?
maybe you got the odd person, the odd microorganism in that
Oh, that's true
community.
too. Yeah, yeah. I was thinking even if you did get one that represented like the data set initially, how long is it until it doesn't because it went on its own path now? But you're right. You don't even know if you got one that was like a normal one or one that
And
represented
even if
more of the
and
average.
even if it were, you've had it in the lab for 20, 30, 50 years.
Hmm.
It's probably evolved into something slightly
Exactly. Yeah.
different.
Is
So
that a
it's
problem that I'm assuming that's a problem that wasn't thought of for like a while. Is that something that's tried to that the researchers try to correct for more now? Like switch out the because I heard someone, it was in a very different topic, but I heard a researcher, it was like on a podcast or something, or maybe even a book, but they were just talking about, oh yeah, it was a much more scary thing because it had to do with humans, because it was about how the strains of like lab mice that are used to study all our diseases are very separate from like normal mice at this point, because they're
mm,
like lab mice, and it had it like they were basically getting to the same issue of a lot of the results from all the data in the past done with these mice to like draw conclusions about what it would do to humans might not actually be accurate anymore, because this mouse is like a different, we've made it a different thing, you know. So it's the same kind
yeah,
of question of how to control for that in research.
yeah, yeah, because sometimes we select strains that are really good at growing in the lab, but that
Mhm.
doesn't mean they're normal.
Yeah.
One of the books that was fascinating for me, which I read when I was at Johns Hopkins, a graduate student there. Um, there's the HeLa line cell. HeLa is Henrietta Lacks cell's lineage. Henrietta Lacks was a Baltimore resident who had a really awful cancer. I think it was somewhere in her reproductive system. I don't remember where exactly. But anyways, she had a very aggressive cancer type, and they collected some samples of her cells without her consent.
Mhm.
And they noticed that they reproduced really well in the lab, so they could do a lot of research with those cells, and it became the HeLa cells. And it is an ethical question now. There's a book about it. I highly recommend for you to read it, and I think they made a movie out of it. It's called The Immortal Life of Henrietta Lacks. And it was very interesting to me because studying biology in Spain, we were always talking about the HeLa cells and how important they were and how we did
For research.
cell
Yeah.
biology with that. And then I found out that it was from an African American woman in Baltimore who had a very aggressive cancer. They took her cells and they started using them for research, and we use them till today.
Hmm. That's
And
wild.
her family didn't get money from it.
Yeah. I think yeah.
So they
The name
told
sounds
us
like, I feel
about
like
that.
I've heard minimal about it, but that's so wild. Yeah.
Yeah, I highly recommend that book. It's really well written and it's a very interesting story. And so they use it
Well,
now to teach us about the ethics. The Immortal Life of Henrietta
oh,
Lacks.
okay. Okay. I was just checking. I wasn't sure if that was just the movie name or also the book. Okay.
Yeah,
Yeah.
the book is what I
Interesting.
read, and I think they did a movie about it recently.
Hmm.
Um, so yeah, that and for COVID, they're also using strains. I was actually hearing about this researcher who was explaining the cocktail that they were using to treat COVID was or came from a fetus. Like again, 20, 30 years ago, there was an
Oh, like the cells they're testing the cocktail against came from like an old cell lineage.
line of cells from an aborted fetus.
Yeah. So yeah, so it's like, I mean, obviously there's the ethics of it, but also just how well does that data represent normal cells that haven't been used for research for that long?
Yeah.
Yeah, that's really wild.
Yeah. So yeah, we still don't know anything about anything. We know so much, but it
Yeah,
still
there's
feels
so much more to go. Yeah, this is it. Yeah.
scratching the surface of it.
Yeah. I recently, like right now, I'm reading it's a book someone gave me a long time ago that I've been meaning to read, but it's a physics like novel book wrote like with and by this one professor at MIT that was just like known for just being really good at describing physics, and it just kind of walks you through a bunch of physics, and it's like beautifully written, and just the whole thing with science in general and physics is kind of like you were mentioning before. You see the world in such a more beautiful way because you're seeing why things are happening the way they do and how it works. Um,
Mhm.
but he was, what was I getting at? What were we just talking about? Um, thing. What did I start talking about before I said that?
Oh, how little we know.
Oh yeah, yeah. Oh yeah. So in the book, he early on he's just talking about the percent of matter that is like the matter we interact with, and it's I think he said it's either 4 or 6%. All the energy and matter that like we know how to see and like understand the world is that, like, I think it was 4% honestly, and the other 96 is like the dark matter and like space and stuff we don't know. So even all this stuff that we're talking about that we kind of know is only in that 4%. And
Yeah.
then it's wild. Yeah.
Yeah, and for the ocean, we still there's so much that we don't
Yeah.
know. Um, it's just so big.
Yeah. Cause well, like 15% of the deep oceans have even kind of been looked at. Like,
Yeah. So one of the things that I'm excited about is there's this project that was just funded with over $50 million by the National Science Foundation to build a robotic fleet of biogeochemical floats. So they're in essence some floating instruments that have several different kinds of sensors that tell us more about the temperature, salinity, which tells us about the currents, and it also measures dissolved oxygen, and it measures pH, and it's all going to give us a bigger picture of what's happening in the world in areas that are hard to sample. Like for example, the Southern Ocean is really hard to sample because there's always clouds, so we barely get
mhm.
satellite images from that
It's hard for
area.
remote
If
sensing.
you go out
Yeah.
on the ship, it's super expensive. A day out on a research cruise is like $10,000. So it's really expensive to go out there. And then um yeah, and then it's hard to sample those big expanses. It can sample under the ice. So we're going to, and in the winter time when there's no sunlight, so we're going to learn a lot more about processes in the ocean that escape our current technology. So I'm very excited about those and how we can combine those with satellites, because satellites also only tell us what they can see on the surface, but
Mhm.
there's three dimensions. So with these
Yeah.
robots, we will be able to understand better what's beneath the
What is
surface.
what's the, so that's a project that's funded through Skidaway?
No,
Those floats.
no,
Okay.
this
What is that called? Like the,
I can send you the link. I just heard about it yesterday through Twitter.
the
But
project of
there
the
was
float drones.
um, They're called biogeochemical floats. And they had a program to study the Southern Ocean that was called SOCOM, and they demonstrated that it was very useful. So now they're trying to do a global fleet.
Hmm. Yeah, that's so
So I'm
cool. That's where my biggest interest lies, honestly, is just the amount of things that can be done in the ocean with drones is like never ending. And
mhm,
that's where I even just because I have, yeah, all my interest is in like the biology side and like the engineering side. I love the all these things that we know will exist someday. Someone needs to create them, and I love that.
mhm.
But yeah, so talking about these kind of drones that can help us in all these answer all these questions that we've talked about is that's the coolest thing to me. I
Yeah.
love learning about all these projects. The last person I got to speak to, he actually also works with like remote sensing and things over in California. Tom Bell, and he works with like kelp, but he let me know
Mm.
about a similar thing I never heard of. Like, have you heard of Argos floats?
Yeah, that's what they are.
Oh, this is a different thing, or this is a different. Is it the same fleet?
It's
Because
the
those
same
ones are
idea.
like going like down, and they're not going
The
across
same
the
thing.
top.
Yeah,
Yeah.
Argo. So these started as the Argo fleet, and they originally only measured temperature and salinity,
Hmm.
which already gives us a lot about just the currents and the dynamics of the ocean. But now they created what they call the BGC Argo, biogeochemical Argo. And this is what this new fleet is. So they added to those chlorophyll, dissolved organic matter, dissolved oxygen. So it gives us more information about the biology and the geochemistry of the ocean, not just temperature and salinity.
Okay,
That's the same thing, but improved.
that's
Yeah.
cool. So they don't function like autonomously yet. Like it's still the same system of we're gonna basically drop a lot of floats and they have like a certain lifespan that they float across like certain depths, but they're not moving themselves. They're not surveying yet.
They just drift with
Yeah.
the current. You don't tell them where to go. There are other robots that you can tell them where to go, and there's one called the sail something, which is like a mini sailboat, sail drone, I think it's called. And there's the gliders. But what we're more interested in now is these kind of floats that drift with the currents. And so you drop them off in different places and they go with the currents.
Mhm.
And like you said, they go down and they take samples as they go down and then they stay for a certain amount of time at a certain depth, and then they come back up, and then they send out the data through iridium, through satellites, and then they go back down again and keep recording, and they do this like up and down motion, but they're always drifting.
Is that
We
why
don't
they're more interested? Is that why they're more desired for research because they get data from like a bunch of different levels in the water column and like depths
it, 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 it
compared to the floating ones? Is that why they're more useful?
no, they all float. Well, the sail drone, I think it's always floating but doesn't go underwater. But the BGC Argo is the same as an Argo, and they go up and down.
Mhm.
And it's good because they just go everywhere.
Yeah.
And they sample at different depths, and they go to places where it's not very easy to get to by
Yeah.
boat or anything. Yeah, so they gave us an idea. It's like it's much better than having a ship in this. Well, it's good and bad. So you don't control where they go, but you kind of know where you might want to drop them because you want to study some process there. Um,
Are there any
yeah.
places they can't get to because the currents won't take them there?
So I know that, for example, they have some issues with the ones in the Arctic Ocean because the water's a lot more stratified, and by stratified it means that there's all these layers of different density, and it's really hard to go up and down, so they can get stuck underneath, and if they yeah, and you can't recover them, and they won't go up and send the data. So I know that's an issue with those systems, but in general, it worked really well in the Southern Ocean. there's also issues as to where they go with the idea of the territorial waters and where you are allowed to
Oh yeah.
take data from.
How does that
Uh,
work? Yeah.
yeah, I don't
No.
know
Yeah.
very well. I went to a workshop and they mentioned that that was an issue because there are some areas of the ocean where we really don't have a lot of data and it's a big gap and we might not be allowed to sample there anyways.
What organization, if any, kind of either rules over that, talks about it? Like, is there any like world body that focuses on like global research? Like global research efforts, like a kind of like a UN for just researchers and data from like global stuff. There's nothing like that.
No, not that I know of. There's big international efforts for different things. So, for example, Argo is one of those. You can have your own float, but if you want to become part of Argo, you have to subject it to certain parameters and conditions so that everybody's doing the same thing.
But
But
safe
then they'll
too.
process data and help distribute it, which is also important.
One of the things you mentioned really early on, just while you were talking, I wanted to ask about it. You talked about like the economic like importance of coral reefs. Could you talk more to that?
Whew. I don't really have the numbers,
Mhm.
but I do know that it supports so many people's, the tourism industry, the fisheries. There's so many people who live because of coral reefs. Australia makes so much money from that, for example, but also state islands, they depend on the corals to make a living.
Mhm.
So it would be a shame if we ignore all these red flags. We know it's
Yeah,
happening, but it is what it is.
yeah. Do we know, and you mentioned a couple times, like, are the three main things that are kind of fighting for, and they're groups themselves, but that are fighting for kind of
Spe-
percentage of those ecosystem? Is it like corals versus sponges versus like
Algae.
algae? Those are like
Hmm.
the main three in any body of water, kind of, other than like, bigger marine animals and stuff.
Well, that's in coral reefs. You
Okay,
don't get
okay. Coral reefs, those are the categories, or those are the biggest categories. Okay,
yeah,
good
yeah,
to know.
yeah. And there, I think there's a lot more sponge cover in the Caribbean than in the Pacific.
Hmm.
Um, the Pacific is has a lot clearer waters. They're more oligotrophic. In the Pacific, there's still oligotrophic, but um, there's more food in the water. They're not as clear, and the sponges like to use up those nutrients in the water.
Hmm. Could you give like in those like three groups, like corals, sponges, algae, um, could you give like a brief comparison, not of like the biological differences of what they are, but just like, like which one produces the most oxygen, which one, um, I'm trying to think of just like the different, like their different like characteristics, just so I can understand the differences better and
Uh,
everybody else. Um, and
I
kind of like
don't
their effects to the environment, I guess.
I don't know that I have the numbers to say something about that.
Uh-huh.
Um, But basically, the coral is the umbrella organism that is responsible for building the reef, the
Mhm.
structure of the reef. And so that's key to build a habitat for fish and for other organisms to live. Without the corals, we don't have a reef. We don't have a structure, a scaffolding structure. Now, the algae are important for most of the fish to eat.
Mhm.
We don't want too many of them that would overkill the coral, and then we stop producing substrate and
Mhm.
structure. And then sponges are kind of in the middle, and they just take up some space to grow, but they can also grow in the sand. There's different species. Some of them can also produce a lot of structure and habitat for fish and for um, they can act as nurseries. Um, but one of the biggest important roles of sponges is that they're continuously filtering the water. So they're cleaning the water.
Hmm.
They're filters. So they're very important to get rid of the excessive nutrients that what make the algae grow really fast, for example. They eat a lot of bacteria in the water. There's also some discussion, and I don't know the exact details about what they actually eat. There's some discussion about that. There's some researchers now that say that they're eating more dissolved carbon. Some people that think it's more the microbes, bacteria in the water, and it might also depend on the species of sponges. That's outside of my expertise, but that's a conversation that's happening right now. But the main thing is this balance between coral and algae, and then sponges are in between.
Hmm.
There are some sponges also that can be problematic for corals and algae to some extent because they bore into the rock, into the calcium carbonate rock, and they grow inside the rock, so they eliminate substrate for other organisms to grow on.
Hmm.
There's that competition, and those can be really aggressive in the Pacific, for example. Um, so that's kind of the gist of it. And like I said, corals are the enigmatic main thing of coral reefs. Without corals, we don't have coral reefs. Um, and what else can I say? And there are some fish that eat coral, but not so much, really. It's more the algae. Um,
Yeah, that really helps get a yeah, that helped a lot to get an understanding of kind
yeah,
of the roles of all three and just kind of the way they interact. Because yeah, that was yeah, my biggest confusion probably before was the differences between the corals and the sponges because I know sponges can build up too. But yeah, I guess the filter component is really I know is really important. Um,
yeah. And corals itself, they build the rock. Sponges do not.
mhm.
Um, and so corals are the key thing there. And it's funny because they grow it underneath them, not on top like other organisms, but underneath them. So they keep building upon.
The
Um,
corals too.
yeah, and then the symbiotic relationship between corals and the Symbiodinium algae that lives within them is really interesting. There's people who have been looking at the different clades and strains that grow inside them and their resistance to heat and stress and things like that. But yeah, just remember that corals, if they weren't, if they didn't have those algae inside, they would be just like jellyfish. They would be transparent, and they would be carnivorous, and they're like upside down jellies. They just eat stuff from the water and filter
Yeah.
stuff.
Would they still make the like would they still create calcium carbonate and like grow without the algae or no? Or just less?
I would think they can still grow, but they're just weaker.
Yeah.
I read somewhere that it was about 80% of their food resources come from the algae. So imagine if
Oh,
you had
okay. Yeah. I didn't know.
to
Yeah. That's a
cut
lot.
down
Yeah. I was
a
wondering
lot.
around where that was.
Yeah.
I read in your or just like looking at your like profile on Skidaway online that one of your interests and it touches on kind of what you'd already mentioned, but like the CubeSats, like the little like the micro satellites. Have you dealt a lot with those, or what do you love about those, and how do they um
Yeah. Yeah.
yeah,
So
everything here?
that's the most exciting thing for me right now. So I told you a little bit of how I went from pure biology to
Hmm.
study marine sciences and corals, and then eventually plankton, right? But the thing that really struck me as important is that we have global problems right now and we need to tackle them with global tools.
Hmm.
So satellites are the perfect tool for this. We can get global snapshots of how much chlorophyll, which is the pigment that the microplankton and that any photosynthetic organism uses to do photosynthesis.
Hmm.
Um, so we can make maps of the chlorophyll in the ocean anytime. Um, and it can become a little bit more complicated, and we can get maps of specific pigments that specific phytoplankton might have. So, for example, if they have a red pigment, like red tide plankton do,
Hmm.
we can see a change in the color of the ocean. It's going to look red. So we can measure the extent of a red tide from space. We can also, we were talking about coccolithophores, they change the color of the ocean to white. We can use that whiteness with an algorithm to make maps of coccolithophores from space
Hmm.
globally. We can also look at from river runoff or the sediments in the water, they're going to make the water turn brown. We can make maps of those river runoff. We can see the extent of an effect of a hurricane, for example, after all that rain happens. That's going to bring river runoff. How is that going to affect the turbidity of the water over the corals, and how long can the
For big,
corals be?
like transparency, like just like
Yes.
how like clear the water is, kind of.
Yes, it's a measurement of how deep the light can penetrate, which tells us about how dark or clear the water is. So we can make maps of turbidity of the ocean around the world. So we can really tackle global problems with satellites. Now, we've had what we call, well, there's many different types of satellites. The ones I'm interested in are what we call the ocean color satellites, and we've had them since 1978. Um, and the first one was the coastal zone coral scanner or something. I'll let you know which one it is.
Hmm.
CCCS. And that one was meant to be for coastal zones, but it turns out it didn't do really well in coastal areas, to our surprise. So it was much better for open ocean. But that was the first time we started seeing the the powerful tool that satellites were. Then there was the Seawave satellite that was launched in 1997, which coincided with one of these El Nino events. And so we suddenly saw an anomaly that we had documented by ship, samples from ship. We saw an anomaly in the Pacific, equatorial Pacific chlorophyll. So that was really striking. We started seeing eddies, which are these whirlwinds in the ocean. Um, we started seeing how dynamic the ocean is thanks to satellites, and it guides us as to where we want to go and sample with the boat and get more detailed information, or with these floats.
Hmm.
But satellites give us that big picture. Um, but building one of these satellites can take a decade or two. It costs millions. You might start off as a junior scientist when they start designing the satellite, and won't see it until you're halfway through your career before the time you see the images. So it takes a really long time. The advantage of CubeSats is that we can build them really fast. They're much cheaper, and the newer ones are even higher spatial resolution. So instead of, if you think about the resolution of the cameras in your phone, if you remember a decade ago, the kind of pictures that it took and the pictures that we can take now, the size of the pixels has really changed and improved. So now we can see things at a much larger scale, or a much finer scale, I should say. So I I've always been interested in satellites, but I don't have a background in engineering. I only use the imagery that was produced by satellites, right? But 2 years ago now, yeah, 2 years ago, I heard of this group who were looking for a postdoc. They had designed a CubeSat, which is a miniature satellite that is the size of a shoebox, is as opposed to a school bus that was one of the last satellites that NASA launched, MODIS. Um, and so they had designed a CubeSat, a miniature satellite, and they were getting ready to launch it, and they needed someone to join the team and help with the management and the interaction between the engineering side of things and the users who
Mhm.
would be using the images, and where we would take the images from and so forth. And so I was like, wow, this is a once in a lifetime kind of thing. I need to apply for this. And so I applied, and I have wonderful advisors that are experts in the satellite remote sensing field, and they vouch for me, and I got the job. And so I've been learning about designing a satellite mission. What do we need the satellite to look at? What are the specifications? What are the wavelengths? What's the orbit? How do we launch it? How do we launch it? We So I joined in June 2018. In December, we were launching the CubeSat with
Mhm.
SpaceX. So I
I
was
was gonna say, yeah,
very
SpaceX helped
lucky
a lot with
to,
all this stuff. Yeah.
yeah. So this was a really neat opportunity, and it was a really interesting partnership between the University of North Carolina, Wilmington, which is where I was based as a postdoc. That's where the principal investigator was. But we also collaborated with the NASA team in NASA Space Flight Goddard, um, and then we also had a Scottish team who built the platform where the sensor was, who built the satellite, whose name is Clyde Space. And then we had another component, which was another investigator, Alan Holmes, based in California, in Santa Barbara, who's he's just amazing. He designed the sensor, and he's designed other ocean color sensors in previous satellite missions. And this is just a dream team of people to work with. And so I've been learning from them how the sausage is really made, how we coordinate with the engineers. I had to learn about things like, oh yeah, the attitude control of the satellite, or the orbit, or the all sorts of engineering terms that I really was not prepared for. But I am very excited that I've learned with some of the best in the industry, and we've started taking some images, and we're still not fully commissioned. Commission is when the mission is ready to start operational, or being operationally ready to produce images for the users on a regular basis. We're still doing some more engineering tests, but the images that we were getting are
It's already
spectacular.
been launched, though. You're already getting data. You just perfect. Yeah.
Yeah. And it was really neat to also work with SpaceX and all that system. And for example, one of the anecdotes that I really liked was that the sensor was built in Santa Barbara. It was shipped to Scotland. From Scotland, they put it together, they did all the tests, and then they had to take it uh, to Seattle,
Hmm.
where all the different CubeSats were integrated into, because we were launching with 64, with,
That's
well,
fantastic.
60 other CubeSats in one rideshare mission. And it was the first time that SpaceX had done a shared mission.
Oh, so you guys were a part of their first, like, yeah, I love SpaceX, so I follow their stuff. as close as I
Yeah.
can. That's so cool.
Yeah, so they had to take it from Scotland to Seattle to do the integration, but we were running late, so they decided they would just take the satellite in the airplane and hand carry it. So it was stored on the overhead compartment of the plane. Crazy, right?
That's
You're
so
carrying a
crazy.
satellite.
Yeah.
Yeah, so they took it there, and then from there it was taken down on a truck to near Santa Barbara from Vandenberg, where it was launched. And so then it was really neat because we were like part of that exciting first time. They really
Yeah.
liked our project. But then the funny part was that um, as they started deploying all the different CubeSats, there were 64, remember, of us?
Mm.
And so they were all emitting a beep, but we didn't know which one was ours. So it took a while until we figured out which one was ours, and then getting everything ready. And the first time we heard it, we were so excited. And that's the other thing. We created a CubeSat club at UNCW. Students who were interested in being part of the program. And so I had them come over for the first time we downloaded an image and the first time we heard it, and they were engaged in the launch preparations and all that. So it's a really interesting opportunity for students in general. I mean, it was super exciting for me, but imagine for an undergraduate when you're like 19 and you're involved with SpaceX and space stuff and
So
satellites.
cool.
It's a big deal. So we were the first ocean color dedicated mission to launch and be um, up in space. But where I'm working at now, the University of Georgia, they also just launched another satellite, which is it's another CubeSat, and it also has the focus of ocean color, but it has a different approach, slightly different approach. So I'm really interested in comparing the two and what different information we can obtain from each one of those CubeSats with different specifications. Um, but that's something that is really the hottest thing right now, building CubeSats. Everybody, universities, government agencies, lots of people are thinking that it's just so much cheaper to build and launch a satellite now that anybody can do it. I'm impressed that there's lots of high school students now working on CubeSat
Yeah,
pro. So one of the things that I want to do now as assistant professor at UGA is create my own hands-on CubeSat building class.
that'd be really cool.
Yeah, so the group who built the CubeSat at UGA, they're amazing. I'm collaborating with them. The director is fantastic, and they were advised by some of the same people I was working with at the Seahawk. Oh, sorry, I didn't say the UNCW project I was working on. The name of the mission is the Seahawk CubeSat, and the
That's the
name
one
of
you guys sent up.
yeah, that's the one that was launched in 2018 with SpaceX.
Mhm.
Um, the one that UGA just launched is called Spock. It is the Spectral Ocean Color CubeSat. And yeah, so we plan on teaching some classes together. There's more engineers there. So they're doing the more advanced course right now, of really designing the mission and everything. But what I'm interested in doing is something a little bit more basic, more 101 for people who are in the environmental science field, who are probably never going to build a big satellite,
Yeah,
but I want them to experience and make it more familiar for them what it means to build something and test it and communicate between the CubeSat and the ground station. It requires some um, computer coding, so they'll learn about that. and then they'll have to analyze the data. So we want some environmental scientists to be there, and or biologists or marine science students, someone with a more environmental background to look at the data. So it's a very interdisciplinary idea of a project where you'll have people who have
So
an
cool.
engineering background, an environmental science, a computer science, math. We need everybody.
It's my
One of the things, yeah,
interdisciplinary stuff is the most so amazing. Yeah, you just want to learn it
yeah.
all.
And I've learned that I really like collaborating with people who come from other fields, because they make you think out of the box, and things that you might never wonder or consider even doing. They're like, yeah, by when do you want this done? I was like, what? That I didn't know you could do this.
Yeah.
So one of the things I'm working on now with Alan Holmes, who was the person who designed the satellite sensor
Hmm.
for Seahawk, the Hawkeye is the name of the sensor. So I told him, I want to make these special measurements of a harmful algal bloom spectral signatures so that I can build an algorithm, but there's no instrument that can do this in the lab. It's like, okay, I'll build you one. So
Yeah, that's so cool.
build a prototype, and so I'm now hoping to get a student at Skidaway who could help me work on
Data.
creating spectral libraries so that we can better identify harmful algal blooms from space.
Do you
So
know how
that's
he went about? Um, did he just build like a specialized sensor that focused more on like a wavelength you needed to?
so the thing is that I wanted something that would have the full spectrum, which is what we call hyperspectral. And so there's one instrument that can do that, but it's for field work, so you need a large amount of water. But what I wanted to do is um, go to the phytoplankton collection that they have at UNCW, that has over 400 species of phytoplankton, many of which are harmful algal blooms, and they have them in little containers, in beakers. And so I need to take a small sample of it. I can't, growing 40 liters of that is just a
Yeah.
lot of food for 400. But if I can just take a small sample, they're all the same species. This is what we call monoculture, single culture. So I wanted to take a small sample of this, put it in an instrument, get the measurement. And so with all those different spectra, I can try to build an algorithm. But
So you were
so what I
taking,
was,
sorry to interrupt. You were taking individual measurements of like like which exact like wavelength is returned when you shine light. Like how what measurement were you taking to then use?
yeah,
Yeah.
so I measure at all the wavelengths in the visible. So we traditionally talk about the photosynthetic active radiation. So the visible part of it that can be used for photosynthesis is typically between 400, which is in the blue region, to 800, which is in the red.
Hmm.
If you go further up in the red, it's the infrared, which we can't see, but it's just the temperature emission. If we go further in the blue, it's the UV, which we also don't see. So I want the part that's in the visible part, but I want all the wavelengths, because all these species are very similar to some extent. If you just use one wavelength, it doesn't tell you anything. You need at least a few.
Hmm.
Um, and even if we say we have 8 or 9 wavelengths, we don't have enough information to distinguish some of the species. But if we have the full spectrum, we're hopeful that we can get more information. It's going to be like a fingerprint. So
Hmm.
you'll see that maybe if the organism is more red, if you do a wavelength versus, say, reflectance here, you're going to see that it's going to peak in the red part of the spectrum, in
And
the
that's
eight.
what it's
So
reflecting.
reflect, when we can measure absorption and reflection,
Hmm.
we typically talk about scattering rather than reflection. Reflection is something else. But so we talk about absorption and scattering processes in the ocean, and that's what creates the end product, which is the reflectance that satellites measure from space. So we are able to measure hyperspectrally the absorption, but there's no instrument to measure the backscattering hyperspectrally. We can measure it at a few discrete wavelengths. And I'm trying to see if by using hyperspectral scattering, we can get at differences between um, species that might have very similar absorption properties. So I'm trying to see if the scattering can give us more information than the absorption, because the absorption is mostly determined by the pigments that they have. And if they share the same pigments because they belong to the same group,
Yeah.
but they have different shapes and sizes, their backscattering might be different. So that's what I'm trying to get at. But there's no instrument to measure
Okay.
hyperspectrally.
Cool.
Yeah.
So what is the back scattering? How does that work?
It's just the scattering, but you can measure it. It depends on where you have. So um, if you just think of it as a light bulb, right? So here's the light bulb, and it's going to start emitting in all directions. You can also have, let's say this is a diatom, this pen is a diatom. When you shine light to it, some of it is going to be absorbed, but some of it is going to be scattered.
Mhm.
And so, depending on the shape, and for example, this one, it varies a lot whether it's
Okay.
horizontal or vertical. It's going to have a different scattering pattern.
Interesting. Yeah.
But if you also take into account the spectrum at which you're shining it at, you can see whether it's scattering more in the blue, in the red, in the yellow, and whatever.
Hmm.
But you can have, when you shine something, some of it gets absorbed, but some of it gets scattered. But you can get it scattered back or scattered forward
Hmm.
or up and down. It can be scattered in many different directions. We typically look at the back scattering. So when you shine the light, what's scattered back, because when you think of it from the satellite perspective, what we use in ocean color is passive remote sensing. You're not shining light at it. You're just looking at how much is being scattered back
Hmm.
from with respect to the sunlight it receives.
Hmm.
We Our satellite's not if here's our plankton and there's a sunlight coming down, our satellite is going to be here. It's not going to measuring what's going this way forward scattering. It's going to measure what's going backward scattering.
Hmm.
So the relationship between what's being scattered back and what's being absorbed versus how much light is being shined by the sun is
Hmm.
what we measure by remote sensing. And then it gets really complicated because the water also absorbs, not just the diatom. The wave crest might affect the scattering. The atmosphere affect is absorbing like 90% going into the ocean and out of the. So in the end, if you think about it in terms of numbers, we only get like 5% of the sunlight back to the satellite.
Hmm.
So we're working with a minimal amount of information, and yet we've done so much with it. It's just
Hmm.
amazing to me.
No, it's super amazing. And it's amazing how we can, like all the things you just listed that we have to like correct for in that data. Like how cloudy was that day? Like what other things were in the water? Um, the even like the angle of the sunlight, like what part of the day it was. So like the angle the sun was hitting it obviously can affect the way the scattering happens, so how much you're getting back upwards. Um, but that's really cool that you could potentially, even if they like reflect back the same kind of wavelength because of the same species, you could potentially, because of the different ways they scatter, differentiate them. That's so cool.
Yeah, that's something that we have to take into account. The shape of the plankton. How is that going to affect the scattering?
Yeah.
That might be very variable. So yeah, it's not easy, but that's something that hasn't been done yet. So that's one thing that I'm working towards.
And that'll just give us much more accurate mapping of what is everywhere on the map,
Right.
of which kind of algae,
Earlier, I was
kind of,
saying,
yeah,
yeah, we can get chlorophyll maps. Great. They
yeah,
have some uncertainties, but we can get global estimates.
yeah,
But we were just talking about changes in coccolithophores, and that's
mhm,
just one of the groups of plankton in the ocean. How are we going to keep track of changes in the community composition of the plankton? We need to fine tune our tools to be able to
yeah,
get down to groups better, if not species.
yeah,
The motivations is harmful algal bloom species, for example, but there's the nitrogen fixing plankton that I was telling you about. They observe at a particular wavelength, and not most satellites don't measure at that wavelength. So we don't have a good idea of how much of it is there. And they also migrate up and down in the water column, so it gets really tricky. But there was, for example, this very interesting study last summer on sargassum, which people often associate with the sargassum sea, right? Well, we were seeing them in the Caribbean, and they were blanketing beaches and destroying the tourism because they rot and they stink, and nobody
oh
wants to
yeah,
be in that beach. So some researchers from U.S. University of South Florida, where there's really good optical oceanographers, they came up with an algorithm to track those sargassum um, in the ocean and try to see where it was coming from. And they were attributing it to changes in Congo. So go figure.
yeah, that's so
But
wild.
yeah, but you have to have a big enough accumulation of sargassum to be able to measure it in several hundred meters, if not a 1,000 kilometer pixel, to be
Mhm.
able to detect them. One of the things I'm interested in is trying to use these CubeSat data, which are recording at 130-meter resolution, try to see if we can detect those sargassum or other big blooms earlier, because our pixels are smaller.
Mhm.
So then we can prepare to warn those countries who depend on tourism to try and get the sargassum before it gets beached.
Yeah, that's so cool. Yeah, you never think about, or I never think about the way that kind of resolution actually like maps onto stuff, like the 130-meter, like each like pixel of it essentially is that much space. That's so cool. It's all accuracy and measurement. That's so
Yeah.
cool.
Yeah.
Yeah, I am really I unfortunately have to go here in like 5 or so minutes, but yeah, this whole podcast episode has been really cool because it really, I'm glad it gave the scope, I think very well, of, in a good way, how little we know, because it's just so exciting, all the different, it really broke down all the things we have yet to learn, and how much we've learned with how little we've been able to access, which I think is super
Mm.
hopeful and great. um but
Yeah,
yeah,
you've got a lot of thing to do now.
yeah,
2
yeah, there's
hours.
a lot. There's so much to go. Yeah. Everyone listening, go after it. Yeah. There's a lot of beautiful problems to solve.